Right, good afternoon everybody. I'm in the fortunate position of trying to make up time, and also the fact that that last question just introduced one of my key concepts, and Mansell introduced you to bacteriophage, so I can hopefully move through some of the more background information and keep us on track. So I'm going to talk to you about mycobacteria, and mycobacterium specifically, or primarily in raw milk. I'll get to the cheese as we go through. But what we've been focusing on in the first instance is looking at for mycobacteria in raw milk. So the title then is Johne's, Crohn's and Beyond. So Johne's disease, for those of you who are involved in the dairy industry, you'll be familiar with as a very debilitating disease that can go through herds and has a great economic impact on the herd. Crohn's disease is a human disease, which I shall explain may or may not be linked to Johne's. The Beyond will be the TB question, which we're not allowed to mention. Right, so just as a little bit of introduction to the background to these organisms. Mycobacteria then, it's a very, very wide group of bacteria. The biology is very, very diverse. But they are characterized by the fact that they are very hardy. They survive for long periods in the environment. But we can split them on sort of a gross level into two groups. And those are the fast growers, which pretty much behave as E. coli in the lab. So we can grow it on media, we can culture it, we get colonies on a plate, and it behaves as sort of a normal bacteria. The slow growers, and I underline it, are 8 to 18 weeks to form a colony. So for some of my PhD students, they put the plates in the incubator, they go away for 18 weeks and come back and look at the result. So it is incredibly difficult to work with. Now this is part of what holds up research into mycobacteria. It's just difficult. It's not something we can easily get hold of in the lab and do lots of experimentation because you lose lots of the samples. So unfortunately, the slow growers include the pathogens, so Mycobacterium tuberculosis, so human TB, Mycobacterium bovis, which is bovine TB, Mycobacterium leprae, okay, for leprosy, and my bug of choice at the bottom there, Mycobacterium avium subspecies paratuberculosis, hereafter referred to as MAP. Okay, so this is the organism that is associated with Johne's disease. Go on, thank you. Okay, so this organism then, it's a slow grower. So it is 12 to 18 weeks. Once we get it going, we can get it down to 12 weeks in the lab, but that's about the best it will do to form a colony. And it has been potentially linked to Crohn's disease. Again, I'll come to the evidence for that in a minute. But the biggest problem we have is that any single contaminant you get into your sample grows faster than your organism. And because they don't grow well in the environment, you can see the media we grow them on there is one of these, it's bright yellow. That's the egg yolks in it. It's nutrient rich. Anything will grow. So you can almost guarantee you will lose 50% of your samples just for the random bit of contamination that got in the tube if you leave it long enough before you've got your colonies. So this is what makes it difficult. And even when we get a colony, all we've got is a colony. That's not specific, diagnostic or selective. What we actually have to do then is a molecular test to know that what we have got on the plate is actually the organism we thought it was. So for this, we use one of these signature sequences. We look for a gene that we know is only found in this group of bacteria, and we PCR it up, copy up that bit of DNA. We get a band on a gel, so I know that that colony has that DNA in it, so it is my organism. So that's the sort of normal diagnostics. So Johne's disease then, for the farmers in the room, it is a ruminant disease, a disease of ruminants, so it's not just cows, but sheep and goat. It's a big problem in Australia in goats. It causes diarrhea, weight loss, reduced reproductivity, and premature death. You get loss, economic loss of the animals. It can be passed horizontally, so from mother to calf, or vertically, horizontally within a herd, animal to animal, or vertically from mother to calf, so it spreads through herds. There is a long period of subclinical infection, a little bit like TB in humans, so the animal can be infected for a long time before you even know there are any symptoms, and this is what makes it difficult to get rid of it from herds because you don't know which ones are carriers. And the estimated cost for the national UK dairy herd is £12.1 million annually. The pictures at the bottom on the left, on this hand side, you can see the sort of emaciated state this poor cow is in. You can see its rear end is very dirty, it's constant diarrhea. The two images of the gut section, you can see that you've got this healthy gut on the left, and the right, this inflamed, its bowel is just lost all its surface structure, so it can't absorb nutrients. So it's a very painful and debilitating disease. So Crohn's, in humans then, has been suggested that MAP may be one of the causative agents of Crohn's disease. This was picked up originally because of the similarities in the disease. So in Crohn's disease then, we have relapsing inflammatory condition of the GI tract. The symptoms are, again, this thickening of the intestinal wall. That's an image of a section that's been removed from a human gut. You can see it doesn't look very healthy. There are ulcers, the gut wall ulcers, ulcerate, so you have to remove those sections. There can be up to 15 bowel movements a day, even if the disease is under control, which means socially it's very debilitating. You get pain, fever. So it is pretty nasty. The scale of the problem then, estimated there are 60,000 Crohn sufferers in the UK, over 500,000 in the US, many more worldwide. So it's a big problem we'd like to address. So, is MAP the causative or a causative agent of Crohn's disease? You can culture MAP from Crohn's patients, and you can detect the DNA by PCR, but you can also can do that from control patients. MAP is an environmental organism. It will pass through the gut. How do you know that what you're culturing is not just been ingested and is going through or is part of a disease? Very difficult to prove. DNA will detect organisms, as we just described. They could be dead. Dead organisms could be a problem because this is an autoimmune disease, so it's very difficult. There is some evidence that if you treat Crohn's patients with antibiotics that the organisms are sensitive to, you can get remission. It doesn't work for all patients. But unfortunately, Crohn's disease is a symptomatic disease. It's described because of the symptoms. So ulcerative colitis, which seems to have a different cause altogether, runs into MAP, so into Crohn's. So when a clinician describes the patient, they don't actually know what actually they've got. So it's very, very difficult to do that sort of epidemiology and that association between disease and an organism. So, what do we do? Where does the balance of probability lie? Basically, what the science has taken to doing is looking at these meta-studies, going back and looking at all the literature and taking the whole body of evidence, not just individual study, does this prove it or not prove it, everything, take it all into account. And where are we? And the bottom line is that every time they do a meta-analysis, it seems to come out with the decision that there is a sufficient evidence to say there is an association. And that's as far as we can go. So it's a concern. We're worried that there is an association. We're not saying it is the causative disease, but it's still something we want to keep under a watchful eye. So, where did we get involved with this? So I come from a food science department background, and the FSA commissioned a study that suggested that 2% of retail pasteurized milk contains viable MAP. So those were detected by culture and PCR. So we knew that those organisms came from pasteurized milk, and we could culture them, and they could be genotyped to say, yes, it was definitely that organism. Now, the 2% number is difficult because it's very difficult to sample milk and get cultures. You tend to lose a lot. So some people say it's an overestimate because you're actually sort of looking for it, and therefore it's difficult to do this science. You haven't got a big enough study number. And other scientists say that's an underrepresentation. But estimates are somewhere that 1% at least, maybe, of retail pasteurized milk does contain this organism. So in the UK, the ACMSF, which is our body that looks at food safety, recommended that it should be eliminated from the food chain, but recognised that at the current climate there was no suitable test for this to be achieved. And so this is where I got involved. So thanks to Mansell, who has now introduced you to the world of bacteriophage. So I say, those of you in the dairy industry are probably very familiar with them because they are a pain in your dairies. But these organisms then are viruses, and they will lead ultimately to the lysis, as Mansell beautifully showed with that animation, of the host cell. So we're working with a particular virus called D29, and D29 is a little bit odd in that it has a very broad host range. It will infect lots of different members of this wide group of mycobacteria. And that's really fortunate for us. So we want to detect organisms, and why would we use a bacteriophage to detect an organism? And first of all, we've come upon this thorny issue of viability. Molecular tests that we often use as a substitute for tracking mycobacteria, because they're so difficult to grow, don't differentiate between live and dead. Now, in a food context, we want to know if it's live or dead, because it makes a difference. Because we know they may have come in with the food, but with the raw materials, but if it's been processed out and is now dead, you know, our food production has been successful. So the presence of that DNA fossil, as it were, doesn't really matter. So, antibody-based tests will detect live or dead cells. PCR-based tests will detect live or dead cells. There are versions of the PCR, as Mansell alluded to, just being introduced, where you can tell the difference between a live cell and a dead cell by PCR. Most commonly, that is done by a process called reverse transcriptase PCR. It is the worst method in the world, even in a really lovely, clean laboratory with a squeaky clean DNA sample to do this. To do it on food is like a nightmare. There's just so many inhibitors and molecules in there. It's not very good. So, when we look at phage, what we're actually asking is a virus to infect a living host cell. And so we need the host cell to be alive to get the virus to replicate. So by definition, most of the phage tests are dependent on the cell being alive. And they replicate very quickly. Mansell showed you go from 1 to 50 within the generation time. So rather than going from 1 to 2, we're amplifying the process of growth. We're getting lots more of our buck back for the one we put in. We're getting many more phage back. So this amplifies the detection event and means we can get more phage, more particles to detect. Now, work we did at the University of Nottingham used this technology of bacteriophage detection, and it's been commercialized initially about 10 years ago as a kit for the detection of human TB. And the company that we worked with over those periods came back to me and said, okay, milk now an issue, can you do milk? So, this is the principle of the test, and I'm going to do this ever so quickly because for the purposes of the presentation you don't really know need to know how this works. But basically what we do is take our sample containing hopefully our bacteria, we mix the phage particles with them and let them start the infection process. Now once those the DNA has been injected inside the cell, the bits on the outside don't really matter. So what we do at that point is treat with a viricide. So any viruses on the outside are killed off, and the only ones that survive have if they've gone into a living cell. And once they're alive, they're in there, they can replicate, and they can come out. Now I need to find them, and this is where the trick that we use here is the fact that this organism can detect lots of different types of mycobacteria. So I add a fast-growing one to the lawn. So I put the bacteria, mix the infected ones with some other mycobacteria that the virus will also infect, and I can see where the virus is killing off those mycobacteria, where it's been protected, because I get a clearing zone. So each of those little plaques is a clear zone where the virus was. So one clear zone, one cell. So that's our equivalence. But this now needs my phage to have a broad host range. It's got to hit the mycobacteria and my fast-growing mycobacteria. It's our map and the fast ones. So the kit originally, as produced, was for MTB in sputum. And in sputum, if you get a patient who comes into a clinic and they're coughing up and you can find lots of TB in their sputum, the chances are they've got TB. So we don't really care what sort of TB it is, we just go ahead and treat. It's just, have you got lots of mycobacteria in your sputum? Okay, we treat you. But now we're in milk, and there's lots of environmental mycobacteria in milk. Okay? So that's now a problem. But what we know is that we can identify the mycobacteria we detect using PCR. That's okay, but that hasn't got viability. That could be a dead cell. If we combine those two, if I find it with a phage and then tell what it is using PCR, I now have both bits of information. I know the cell is alive, it protected my virus, allowed the virus to grow, and I can go in, test the DNA and say, was it MAP? So that's our summary of our assay. We take a sample, we mix it with the phage, we plate it out, we get our plaques. In that center of that plaque somewhere was the cell I originally infected. We extract the DNA from that and we PCR so we know what we have found. So, sampling cheese. That was another area of difficulty. So, the problem with the cheese now is it's a three-dimensional, fairly complex matrix, and the mycobacteria are in there somewhere. So we went through a lot of work, developmental work, basically just working on ways of getting the mycobacteria out of the cheese so we could detect them. So it involved homogenization and centrifugation and filtering, but we came up with an assay that would work. And we carried out a survey of about 100 different cheese samples from retail outlets in Cyprus, because the student I was working with happened to be from Cyprus. We did hard and soft cheeses, cow, goat, sheep, mixed milk, pasteurized and non-pasteurized milk. And basically, we found nada, no plaques, okay? So, the PCR result was saying lots of MAP in this cheese. If you do the same method, testing just by PCR, lots of MAP. Plaques, nothing. So it suggests that the production is killing off the MAP. Okay? So this is good news for the cheese industry. Okay, this is very preliminary, small study. Do not hang anything on this yet, okay? But the first indication, now this agrees with older data that was done by culture, but the culture data is always questioned because of all this problem of losses and sampling and all the rest of it, okay? So we published that one last year, and we're just about to go out now with the second paper, which is going to do the technical review. Now what people want to know is, okay, you've got this test, how well does it work? Is it real? So this is the milk data where we've really taken it apart. Now this is the same cows that produced all of that milk in Cyprus. So we were sampling every herd in Cyprus. And these are our results. So first of all, just to show you that despite the homogenization, the filtration, and all of the centrifugation steps, it is remarkably reproducible. So 2 independent 50 ml samples separately processed and sampled from the same animals, and you can see we get a nice correlation between the numbers. So if it was perfect, you get a diagonal line up the middle. A Pearson correlation of 0.94 is the maths way of saying they're pretty good. It's pretty reproducible. But what we were seeing was milk contains mycobacteria. So all the green ones at the bottom left as you're looking at it, those ones were PCR negative. So I've got viable MAP, but they are, viable mycobacteria, but they are not MAP, they're other mycobacteria. But what we saw was all of these ones going up, the high numbers, these ones were coming up with para-TB. Now what was bizarre is there is no para-TB in Cyprus. No cases, unknown, totally. No clinical signs, their bulk milk is full of it. So, the question we were asked by the vets was, well, how do you know that this is not getting into the milk from an environmental source? It's nothing to do with your milk. You're milking cows, you know, it can get in. So what we've plotted here is the TVC, the bulk number of bacteria against the presence of mycobacteria. There is no correlation. You can have high TVC and low Mycobacteria, or low Mycobacteria, low TVC and high Mycobacteria. It doesn't look like it's coming from an external source because it's just a random scatter of data. If we stratify the data and look now at, I split my samples into the ones that have got para-TB and the ones that haven't. You can see all the blue ones are the environmental Mycobacteria. Okay, the TVC is going up that axis, so you can have from low to high, and you will have a random number of Mycobacterium, all pretty low, around less than 50 cells. Those animals that are infected, you can see the other group there, you're suddenly getting this scatter of Mycobacteria 50 upwards. So the number of Mycobacteria in the milk was saying that suddenly, with no external source, the number of Mycobacteria going up, they are para-TB, these are coming from the animals. These are being directly shed into the milk. So, I said it would be brief because this is where I wanted to sort of draw a vein over where we've got to. We now believe we have a practical assay that we can use for routine testing of milk and cheese. So if you're looking at the quality of your milk, maybe TVC is an indicator, but as we've heard, maybe there are more tests that we would like to know. Are there Mycobacteria at high levels in your milk? And also, we find that the plaque data for raw milk indicated far higher prevalence than is reported by the culture method. So when we cultured our samples, only 2 of them were positive. And yet we found para-TB in lots of samples. Now, worldwide, there have been lots of studies on the prevalence of Mycobacteria, and using molecular methods, it says it's really prevalent. And all the culture people say, no, it's not, we can't find it. Well, our data says that the PCR is right. There is a lot of hidden Mycobacteria infection in the world. So, our data supports the fact that the prevalence estimates, the high end, are correct. And that is a concern for those of you using raw milk, because we now know that we've got lots of asymptomatic animals where they don't know they've got Johne's disease that could be introducing into the milk. The good news is the preliminary data says your cheese is killing it. So we've yet to go further with that. But then let's go to the thorny question that we're not allowed to mention. Our test detects Mycobacteria. The PCR test tells me whether the plaque is due to para-TB or another type. But I can modify that PCR test. I can look for other signature sequences, and we've done that. So in this test, we have got a PCR assay that says, is it other Mycobacteria, which is lane 3, lane 4, that lane slipped, okay, lane 4, this one here. That tells me I've just got the environmental Mycobacteria present. If I have a paratuberculosis, I would get this band signature. If I have TB, I get that band signature. So we now have one test that we can apply to milk samples to ask the question whether or not we have para-TB or TB in a milk sample. And that result takes 48 hours. Okay, so there we have rapid quantitative tests developed for detection of MAP in milk and cheese. We have improved sensitivity, we believe, over traditional culture methods. It does provide live-dead differentiation. The DNA in our assay is therefore preserved for genotyping, and it is equally applicable for detection of bovine TB, and it would tell you whether that bovine TB is inactivated or not. So, I'd like to thank the team, which was George, who came from Cyprus to do his PhD with us, is now back there teaching in the Cyprus University, but still collaborating with us. The current crop of students, and John Huxley is my friendly vet who finds cows with Johne's for me. So, thank you very much.